everyone, this is Group 1's presentation on Draculin, presented by Yilin, Michael Doan, and myself, Julia Martinet. A quick note before we begin, in parts of this video, we're going to be talking about blood, and we'll have some pictures with blood in them. If that makes you feel queasy or faint at all, please take precautions so you don't get sick.
Draculin was first discovered on October 1998 by scientist Anna Z. Fernandez and her fellow scientists. They found anticoagulants activity in the vampire bat's saliva, which prevent the victim's wound from healing while the vampire bat is drinking its blood. It was not until 2003 another study proved that Draculin can be used as an anticoagulant. As of October 2020, Draculin-based drugs is only undergoing phase 2 of clinical trials and have not yet been approved by the FDA. Now let's learn more about the vampire bat. Only 3 of more than 1,300 other types of bats are vampire bats. The common one, Desmodus rotundus. The white wings, Thymus youngus. The hairy legs, Diphilia equida. The one we're going to explore more is the common vampire bat, which is the one that has Draculin. It is found in the regions of Mexico, Central America, and South America. They are only 9 cm long with an 18 cm wingspan. They weigh around 25 to 40 grams. They are social creatures, meaning that they are usually gathering in colonies about 100 to 1,000 animals. They are often considered as a pest because they can spread rabies to livestock.
Now to why common vampire bats are considered so spooky, their feeding habits. While they do prefer large mammals, common vampire bats rarely, if ever, feed on human blood. They mostly drink blood from livestock like horses and cattle, or native prey animals such as tapirs and capybaras. Like other bat species, these bats use echolocation and their keen sense of smell to detect a prey animal. Once they find one, the bat will land from above or jump up from the ground onto the animal's neck, flank, or rear. Once the bat has found a blood vessel close to the skin surface, they bite through the skin and begin drinking using a grooved tongue. An anticoagulant protein in the bat saliva, Draculin, helps to keep the blood from clotting before they finish feeding. Over the course of the meal, the bat may have consumed up to 20 grams of blood, half its body weight. While the thought of drinking blood to survive is certainly creepy to many of us, it's actually very rare in animals. One reason is that blood is very nutrient poor. It's basically water with a little bit of salt thrown in and some other things. For the common vampire bat, this means they have to consume a huge amount to meet their nutritional needs, and this is a real problem. If they drink too little, they don't have enough energy to get back home, but if they drink too much, they're too heavy to fly.
Through studying other anticoagulant proteins, drugs have been developed to specifically target anticoagulant issues. By definition, anticoagulants are chemical substances that prevent or reduce coagulation of blood, thus prolonging the blood clotting time. Coagulation, or clotting, is defined by a process which blood changes from a liquid to a gel, forming a blood clot and therefore stopping bleeding. Examples of anticoagulants are warfarin and heparin. Warfarin is a medication that is used as an anticoagulant. It treats blood clots, mainly deep vein thrombosis and pulmonary embolisms. In addition, warfarin can also be used to prevent strokes in people who have aortal fibrillation, valvular heart disease, or artificial heart valves. Heparin is a medication that is used also as an anticoagulant. It mainly treats heart attacks and unstable angina by injections. In a similar manner to how warfarin and heparin were developed by studying other proteins that cause anticoagulation, it is hopeful that Draculin can be used in a similar manner.
Vampire bats are only one piece of the larger natural product anticoagulant puzzle. Many other hematophagous or blood-eating organisms have anticoagulant compounds in their saliva. For example, many blood-sucking leeches have an anticoagulant protein called hirudin that was used to develop several direct thrombin inhibitor anticoagulants. Studying the saliva of these organisms can also help researchers gain a greater understanding of how blood clotting occurs. Other lead compounds have come from plant sources. Coumarin is found in a wide variety of plants, including tonka beans and sweet clover, and was the lead compound for a class of anticoagulants called vitamin K antagonists, which includes warfarin. Some anticoagulants are derived from animal tissues with little to no modification. For example, heparin is isolated from pig intestines and cattle lung tissue, and it's used as a short-acting anticoagulant. It's a polysaccharide, and it's normally found in the mast cells of many vertebrates and even some invertebrates. Its usual physiological function is thought to be defense against foreign pathogens rather than straightforward anticoagulation.
For background information, Draculin is a glycoprotein found in the saliva of vampire bats. Draculin is composed of 411 amino acids and works as an anticoagulant. As an anticoagulant, Draculin inhibits coagulation factors 9 and 10, which results in the increase of blood flow. Coagulation factor 9 is one of many serine proteases of the coagulation system. A deficiency in coagulation factor 9 generally results in hemophilia. Coagulation factor 10 plays a key role in hemostasis. It is a central part of blood coagulation cascade, which catalyzes the production of thrombin and leads to blood clot formation and wound closure. It is an attractive target for the development of new anticoagulant agents. A deficiency in coagulation factor 10 is very rare. About 1 in 1 million people can develop this defect. Side effects include nosebleeds, joint bleeding, and gastrointestinal blood loss. Direct coagulation factor 10 inhibitors are very popular anticoagulants. Under normal conventions, when bleeding occurs, the calcium channels are open, which causes an influx of calcium ions. With calcium ions in the body, this causes a reaction leading to the vasoconstriction of smooth aortal muscles. This in turn decreases blood flow and therefore stops the promotion of blood flow and prevents death. However, when Draculin is introduced, it in turn activates the CGRP receptors and opens up the potassium channels. This in turn will decrease the calcium ions within the smooth muscles, causing it to relax and promote continuous blood flow. This continuous blood flow will allow the vampire bats to enjoy their blood-feeding meals. So now let's talk about how Draculin works. When the vampire bats bites into their victims, they puncture a hole into their victims, which allows saliva containing Draculin to enter the victim's body. Blood flow is increased because DSPA increases the lag phase of the coagulation factors 9A and 10A. In addition, the inhibition of coagulation factors by Draculin prohibits the conversion of prothrombin to thrombin and the conversion of fibrogen into fibrin, which therefore reduces the coagulation of blood flow. As a result, Draculin keeps the blood flow of vampire bats' victims due to the prevention of blood clotting. The mechanism of action for Draculin is described as activating the CGRP receptors and potassium channels to relax the aortal smooth muscles of the incision site to release blood flow in a continuous manner for blood feeding. Using Draculin results in blood thinning to prevent coagulation. In addition, Draculin's biological function pathway is reversible because it acts as a non-competitive inhibitor. As a non-competitive inhibitor, Draculin may be useful in anticoagulated treatments in diseases such as strokes, heart attacks, or deep vein thrombosis. Now, let's understand why Draculin is important to humans. Scientists have determined the amino acid sequence of Draculin is very similar to human CGRP and also rat CGRP. Down below are the amino acid sequence of these proteins. Highlighted in green are the differences in the CGRP amino acid sequence between vampire bats, rats, and humans. As shown here, there aren't that many differences between their amino acid sequence, and therefore studying one specific protein can provide some insight into the other proteins.